Hello students, this is Mrs. Sonal Patil. In our last class, we had begun chapter 6, Molecular Basis of Inheritance, in which we covered DNA, features of three-dimensional double helical structure of DNA, and we have studied about packaging of DNA. Today let us begin with the transforming principle. Firstly, we will discuss about the search for genetic material. Even though the discovery of nuclein by Mischer and the proposition for principles of inheritance by Mendel were almost at the same time, but that the DNA acts as a genetic material took long to be discovered and proven. By 1926, the quest to determine the mechanism for genetic inheritance had reached the molecular level. Previous discoveries by Gregor Mendel, Walter Sutton, Thomas Hunt Morgan, and numerous other scientists had narrowed the search to the chromosomes located in the nucleus of most cells. But the question of what molecule was actually the genetic material had not been answered. Transforming principle. Students, remember that this question is very important from exam point of view, may be asked for 5 marks. Frederick Griffith, a British medical officer, described the phenomena of bacterial transformation. So Frederick Griffith in the year 1928 conducted series of experiments with Streptococcus pneumoniae. This Streptococcus pneumoniae was a bacterium responsible for pneumonia. He witnessed a miraculous transformation in bacteria. During the course of his experiment, a living organism had changed in physical form. When Streptococcus pneumoniae bacteria were grown on culture plate, some produced smooth shiny colonies and others produced rough colonies. The one which produced smooth shiny colonies were called as S strain because they had mucus coat, and because of which they were called as S strain, while R strain did not have the mucus coat. So what Griffith did was that he injected S strains into the mice and observed that the mice died of pneumonia. So mice infected with S strain, which was virulent, die from pneumonia infection, but mice infected with the R strain do not develop pneumonia. Nextly, what he did is that Griffith was able to kill bacteria by heating them. So what he did, he killed the bacteria, he heated them and killed them. He observed that heat killed S strain bacteria injected into mice did not kill them. But when he injected heat killed S and live R bacteria, the mice died. Moreover, he recovered living S bacteria from the dead mice. So firstly, what Griffith did was that he injected S strain into the mice, which was virulent. Mice died of pneumonia. Secondly, what it is that he injected R strain into the mice, which was non-virulent. Mice survived. Thirdly, he heat killed those S strain bacteria and injected them into the mice, but the mice survived. And lastly, what he did is that he mixed, or he heat killed S strain bacteria and mixed with R strain, that is live bacteria. Heat killed S strain was mixed with R strain with live bacteria and injected into the mice, and he observed that the mice died of pneumonia. So by using S strain, that is heat killed, and R strain, that is live, it was concluded that R strain has been transformed by some material of S strain which makes R strain virulent and unable to synthesize smooth polysaccharide. This must be due to transfer of genetic material from S strain to R strain. So Griffith concluded that the R strain bacteria had somehow been transformed by the heat killed S strain bacteria. Some transforming principle transferred from the heat killed S strain had enabled the R strain to synthesize, as I told you, it was unable to synthesize a smooth polysaccharide coat and become virulent. This must be due to the transfer of genetic material. However, the biochemical nature of genetic material was not defined from his experiments. So this was about the Griffith experiment. Once again, diagrammatically it has been shown here in this slide. So we can see here rough non-virulent strain, R strain, injected into the mice. Mice was healthy. Smooth S strain bacteria, when they were injected, mice died of pneumonia. Thirdly, Griffith, what he did, he used heat killed smooth virulent strain. Mice was healthy. And when he observed that rough non-virulent R strain was mixed with heat killed smooth virulent S strain bacteria, when injected into the mice dies, and live strain S bacteria in blood sample from the dead mouse were being observed. Nextly, coming to the establishment of biochemical nature of transforming genetic material. So this is a biochemical characterization of transforming principle. They came to know that by Griffith's experiment, he came to know that there was some substance which was responsible for the transformation, but it was not known which was the that particular substance. So now, prior to the work of Oswald Ivory, Colin MacLeod and McCarthy, the genetic material was thought to be a protein. They worked to determine the biochemical nature of transforming principle in Griffith experiment. They purified biochemicals, that is proteins, DNA, RNA, etc. So they purified these biochemicals from the heat killed S cells to see which ones could transform live R strain or R cells into S cells. They discovered that DNA alone from S bacteria caused R bacteria to become transformed. They also discovered that protein digesting enzymes, that is proteases, and DNA digesting enzymes, that is RNases, did not affect transformation. So the transformation substance was not a protein or RNA. Digestion with the DNAs did inhibit transformation, suggesting that the DNA caused the transformation. They concluded that DNA is the hereditary material, but not all biologists were convinced. So here, what these scientists did is that Oswald Ivory, MacLeod, and McCarthy, they in revealed the chemical nature of the transforming substance to be DNA. They purified DNA, RNA, protein from heat killed S strain to see which of them could transform R cells into S strains. So they observed that protein digesting enzyme, that is proteases, and the RNA digesting enzyme, that is RNases, have no effect on transformation, whereas digestion of RNA with DNAs would not cause transformation. This finding established the transforming genetic material is DNA. The genetic material is DNA. It can be shown by the Hershey and Chase experiment. The unequivocal proof that the DNA is a genetic material came from the experiments of Alfred Hershey and Martha Chase. They worked with viruses that infect bacteria called bacteriophages. So bacteriophages are nothing but they are the viruses which infect the bacteria. These bacteriophages attaches to the bacteria and its genetic material then enters the bacterial cell. So when the bacteriophage attaches the bacteria, the genetic material of the bacteriophage enters into the bacterial cell. The bacterial cell treats the viral genetic material as if it was its own. So the bacterial cell will treat that the genetic material is its own, the viral genetic material is its own, and it starts subsequently manufacturing more virus particles. Hershey and Chase worked to discover whether it was protein or RNA from the viruses that entered into the bacteria. So they wanted to know whether it was protein or DNA which entered into the protein DNA which entered from viruses entered into the bacteria. So what they did is that they grew some viruses on a medium that contained radioactive phosphorus and some others on the medium that contained radioactive sulfur. Viruses grown in the presence of radioactive phosphorus contained radioactive DNA but not radioactive protein. So remember, viruses which were grown in the presence of radioactive phosphorus, they contained radioactive DNA but not the radioactive protein, because DNA contains phosphorus but protein does not. So remember, DNA contains, we have seen in the DNA structure, phosphorus is present. So phosphorus is present in the DNA but it is not present in the protein. So protein does not. Similarly, viruses grown on radioactive sulfur contained radioactive protein but not radioactive DNA, because DNA does not contain sulfur. Radioactive phages were allowed to attach to E. coli bacteria. Then, as the infection proceeded, the viral coats were removed from the bacteria by agitating them in a blender. So agitation was being performed, it was performed in the blender so that the viral coats were removed. The virus particles were separated from bacteria by spinning them in centrifuge. Bacteria which was infected with viruses that had radioactive DNA were radioactive, indicating that DNA was the genetic material that passed from virus to the bacteria. Bacteria that were infected with viruses that had radioactive proteins were not radioactive. This indicates that proteins did not enter the bacteria from the viruses but it was DNA. So DNA is therefore the genetic material that is passed from virus to bacteria. So that was about the Hershey and Chase experiment. A very important question again from exam point of view. So remember, please, Griffith experiment is important for 5 marks. Again, Hershey and Chase experiment is also important. So please go through this and be perfect with these. Diagrammatically it has been shown here, the Hershey and Chase experiment. Please practice the diagram along with the description. You have to explain this in the examination. So first step is infection, then blending, and third step is centrifugation. So today we will stop here, and in the next class we will begin with the next part of the chapter. Thank you.